Right, we've looked at how DNA can replicate itself and create an identical copy, preserving this code of A, C, T, G, etc., etc. Okay. Remember that C can only join to G and A can only join to T. Okay, this is important. We're going to look at something today called transcription. Transcription. Okay. What you need to remember is every cell in your body that's doing something active has got a copy of all the genes, okay, apart from red blood cells and egg and sperm cells. We'll forget about those. But every cell that's doing something has a copy of the whole genome, okay? But certain cells only want to read certain parts of it. So you can think of your genome as a library, and every cell has got the same library in it, but each cell only wants to read a certain part of it. So how does it do that? What it will do, you've got your genes all coiled up, and you've got this, let's say you've got this sequence here. This is a bit here of gene. And let's say this is a sequence of genes, okay? And remember, I said that the genes are like letters. So you've got A, C, G, okay? And every 3 letters forms like a word that we call a codon or a triplet, okay? A, C, G, G, C, C, A, T, etc. I'll do that. So here we've got 3 words, okay? That might be part of the gene for something. Let's say you can have anything from 20 or 30 up to several hundred or even 1,000 of these codons together. All of that together would form a gene for something, okay? And there are 20, 30,000 genes in the human body. But let's say a particular sequence of about, let's say, a couple of hundred of these triplets will form a gene. Now, this particular cell, let's say it wants to read this. What it needs to do, it needs to open up this. So what it will do, it'll take this part of the gene here, and it will open up, okay? If it needs to make a copy. It's a bit like going into your library, photocopying the page of the book you want and taking it out and using it. And that's what transcription is all about, okay? What will happen is the piece of the gene will open up like this, okay? This might be your code that you want on here. A, T, C, G, C, 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 T, A, G, etc. It will open it up, okay? Where you've got the opposites. You've got T, A, G, 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 A, T, C, okay? Now, floating around in around here, the same way DNA replicated, you've got these base pairs, but they're made of something called RNA, which is slightly different than DNA, but it has a very similar code, but not a completely exact code. And what happens is the A's will line up on the A's like that. You get an A. The C's will line up on the G's like that. But there's another one. So over here, this is the code for DNA. For RNA, you still get C and G, but you still get A, but the T has been replaced with U, uracil, okay? Thymine has been replaced with uracil in RNA, okay? So here you would get a U, okay? And that starts to make a strand of something, okay? So that makes this makes a copy, and you'll see it's the same as that side. This is called the sense side, okay? So this is now called RNA. This is messenger RNA, mRNA, this strand here. And this strand is what the body then uses. This is like your photocopy. It's a bit like a photocopy of that side there, but they've just replaced the T's with U's. So this would be A, U, C, and this is messenger or mRNA, and this is the bit that you've photocopied, and this goes on, and the body uses this in the next process, which is called translation. And translation will turn this into a specific protein. And it's a protein which is the enzyme or the structure or the thing that your body wants to make and helps it do things.